Hello madam, I'm HEERANYAA NADARAJA and I'm from DPT 4A. Today I would like to perform on simulation of gel electrophoresis. Before entering to our simulation, let's talk about gel electrophoresis. The gel electrophoresis is a technique that used to separate the biological molecules. In the simulation, gel electrophoresis will be also used to separate the dyes and visualize them in an agarose gel. Agarose gel when an agarose gel is melted and then cooled in aqueous solution, hydrogen bonding causes it form a gel. Under the influence of an electric field, a population of DNA fragments generated by restriction enzyme will move through an agarose gel, whereby negatively charged DNA molecules will be drawn to anode. Their mobility is almost determined by size, with the largest molecule having the lowest mobility and the lowest molecule will having the largest mobility. The pore size is determined by the concentration of the agarose in the gel, and a DNA fragment of a specific size will migrate at different rates through gels of various concentration. Let's start the stimulation. Start stimulation. We will conduct a gel electrophoresis to separate different solution of dyes. Here we have 5 important points for us to get to know before this laboratory starts. The gel electrophoresis is usually used to separate the biomolecules such as DNA and RNA in the agarose gel with the tiny spores according to their size and electrical charges. An electrical current applied to the gel migrate the charged biomolecules towards the oppositely charged electron. Since the DNA fragment have a similar negative charge, they all will move towards the positive electron. Smaller molecules will move or will migrate through the pores in the gel faster than the larger molecules. Loading dye is often used to visualize how fast a gel is running and add weight to your sample, allowing it to sink to the bottom of the well. Here we have it. It's the wells always be with the negative electrode. It be close with the negative electrode, and then once we place the DNA fragment into the wells, it will be migrate to the positive electron because the DNA fragment contains negatively charged, so that it will migrate to the positive electron. So here, if it's near to the well, it will be a large molecule. If it's in middle, it will be the medium size molecule. If it's near to the positive electron, which means far away from the well, it will be small molecules, which means the DNA fragment is smaller. Now, I would like to introduce the materials and the apparatus that we are going to use for this simulation. Here, first, materials. Here we have some reagents to use for this experiment. S1 solution is the tube containing a dark blue liquid. Same goes for S2 solution and S3 solution. 3 of this solution contain the dark blue liquid. And here we have 1 times sodium bronate buffer, which is a salt containing buffer at 1 time concentration to cover the gel and transmit charge through the gel, allowing the electrophoresis to occur or to process. And here we have the apparatus. Here we have various type of micropipette such as P2 pipette, P20 pipette, P2000 pipette and P1000 pipette. Usually this P2 pipette used for the volume of the liquid between 0.2 to 2 microliters. This P20 micropipette, we will use the volume of liquid between 2 to 20 microliters. This P200 pipette we use for 20 to 200 microliters, and this P1000 pipette we use for the volumes of a liquid between 2000 to 1000 microliters. And here we have the tip box for the particular micropipette. Furthermore, here we are having some electrophoresis equipment such as agarose gel, which is used to be placed into the electrophoresis box to help the sort of the molecules by the size and charge. Here we have the gel electrophoresis box, which is used for conducting the gel electrophoresis, has the anode and the cathode concentration point, which means the positive and negative electron. And then here we have the power supply with the red and black cords, which must be connected to the connection point to on the power supply. And here we have the power supply leads, which are in two colors, which are black and red. Furthermore, here we have some other equipment such as microcentrifuge, which helps us as a motor-driven centrifuge that spins the sample at a high speed to collect the liquid at the bottom of the tube for easy transfer. And then here we have the trash, or we can also known as a biohazardous box also, for us to eject the tip of the micropipette after the use. Let's perform on the procedure of the gel electrophoresis simulation. Here we have the 6 procedures for us to perform. First, we have to prepare the electrophoresis box, then centrifuge the solution tubes, draw up the solution 1, pipette the solution 1 into the well, and then we will be continue this step for solution 2 and solution 3. Then pipette other solutions into the remaining well, as I said. Conduct the gel electrophoresis. Let's stimulate. Select the agarose gel, then place the agarose gel into the electrophoresis box. Orient the well gel toward the negative electrode. This is the negative electrode, this is the positive electrode, so we need the well to be near to the negative electrode, so we have to rotate. Pour the flask with the 1 times sodium borate buffer over the gel box. Place it. Next, select the microcentrifuge to open the lid. Select and move each of the solution tubes to the microcentrifuge. These are the solutions which known as S1, S2 and S3. We are keeping the solutions into the microcentrifuge. And then make sure the solution should be balanced inside the centrifuge. Here we go, we got the balanced ready. So close it. Close the lid. Then press the button to spin at the bottom of the solution. Just open the lid of the microcentrifuge, then remove the tubes from the microcentrifuge. Then select the microcentrifuge to be closed. Next, make a note to show which solution is my pipette with each land. Then select P20 micropipette. Set the volume for 10 microliters. Save the volume. Then select the P20 tip box. Attach it to the tip box. Close the tip box and then select the S1 solution to open and place the micropipette into it. Draw up the solution and then close the S1 solution. And then next, the pipette solution 1 into the well 1. So we are moving it into the gel electrophoresis. Load it in the well 1. Next. Press. Done. And then after finishing the procedure, we have to eject the tip. Next. Repeat the following step for the solution 2 and solution 3. S2, draw up, close. The S2 solution should be into the line 2. Done. Eject the tip and then do the same for solution 3. Eject the tip. Next, we place it here. Cover the gel electrophoresis. Then select the red and black cable and connect them to the red and black connection point on the power supply. And then on the lid of the gel electrophoresis. Okay, just connect it. This is the black cable that we are connecting now. And here we have the red cable to connect it. Oh, sorry. Should be here. Should be here. Yeah. Then turn on. Then set the voltage to 130 voltage. So it should be 130. Okay. Then set a timer for 10 minutes. And then start the timer on the power supply. Let's wait until after 10 minutes for the 10 minutes. Then turn off the switch and then unplug the electrodes from the power supply, from the gel electrophoresis and the power supply. Okay. Then carefully remove the cover from the gel electrophoresis box into your gel. Next. In this simulation, we are using the gel electrophoresis to separate the 3 different dyes. So the 3 different dyes are probably different in size, with the yellow dye will be being the smallest in size, the blue dye is being the larger in size, and the purple dye is being the largest in the size. So if the blue dye is smallest in size, it may migrate faster and be close to the positive electron. So it will be in third place. If the blue dye is larger, it will be in medium state in size. Here we will have the DNA ladder, right? So the DNA ladder will show us the size. Then the purple dye is being the largest one, so it only migrate a bit, like just a bit only. So this is my prediction. Let's see how correct is my prediction. After the procedure that I did, here I had the actual result of this simulation. This is my predicted result and this is my actual result. Here I predict that the yellow dyes will migrate faster and farther. The blue dye will migrate quite medium level, and then it stays in the medium. And then the purple dyes will migrate slowly, and there's only least distance between the well and the dye. But my prediction was quite wrong, because here it shows the yellow dyes is the fastest and farthest from the well, but the blue dye is the slowest and there has a least distance. The purple dye was in the middle. In the middle. Here we go. This is the ideal result. This is because not only based on the size it decides the distance. It's also based on the charge in the DNA fragment. Here we have the clear explanation. We was expecting that the blue bands will migrate farther than the purple dye. Yeah, I also was expect that the blue dye will migrate farther than the purple dye. But it was not possible, because in practical the larger purple band moved farther than the smaller blue band, because not only in the size of the fragment, also its charge and determine but it also its charge determine how far it migrate in a gel, and the purple dye has the greater negative charge per mass than the blue dye. As result, it migrate the farther than the blue dye in the gel. So here we go. So when all the molecules in the sample have the same charge to mass ratio, such as a sample of the DNA fragment, the only governing how far they migrate in the gel is their fragment size. Well, that's all from me. Thank you, madam.